So first, let's talk about the point of meiosis. So meiosis, the purpose is not to make new identical cells. You're not rebuilding damaged tissue or anything like that, or replacing old and dead cells. Instead, meiosis is specifically about creating sex cells. So, sex cells. And there's 4 daughter cells after meiosis because there's one round of meiosis involves 2 rounds of division. There's meiosis 1 and meiosis 2. So if cells divide twice, you end up with 4 cells at the end of meiosis. So by the way, sex cells include sperm and egg. And each sex cell is genetically unique. And that is the most important part of sexual reproduction. Not all organisms reproduce sexually. A lot are clonal species. A lot of species on this planet are clonal species, but sexual reproduction, since its evolution, has really taken off. And the reason is there's a huge benefit to having diversity in a population. So if every individual is genetically unique instead of a clonal copy of the parents, that provides the species with a lot of resilience to changes in the environment. And as we know, the environment's always changing. I mean, in addition to the Earth going through these cycles, these warm and cold cycles, and having these broader scale climatic changes, there's also natural disasters, so like volcanic eruptions that completely change the surrounding environment, or hurricanes, or fires, or things that dramatically change the landscape in which an organism lives. There's also diseases that come through. There are invasive species that come and go. So all of those things dramatically change the environmental stresses that a species is exposed to. So the problem with being a clonal community is that when the environment changes, if nobody has the genes that provide some resilience, some ability to survive in those new conditions, then the entire species will die out. But if every individual in the species is genetically unique, the chances that at least somebody in there is going to have genes that allow survival in those new conditions, the chances that somebody has genes that enable it to survive increases if everybody's different. Everybody has different strengths, everybody has different weaknesses. So some individuals do well in certain environmental conditions, but then the environmental conditions change and other individuals can do well. So having diversity really bolsters a species' ability to survive in an ever-changing environment. So the way we get diversity is through sexual reproduction and through meiosis, which is the production of genetically unique sex cells. Another thing that's important about these sex cells is that they're haploid. So all of our cells are diploid except for our sex cells. So diploid means that there's 2 sets of genetic information, and I've shown that in the cell that we're going to start with here. So each of you, so let's just make this personal, make this about me. So every single one of my cells, my somatic cells, my not sex cells, has a copy of every chromosome from my mom and a copy of every chromosome from my dad. So we'll say that mom is black, dad is blue in this. So I have 2 of each chromosome. So we've already talked about sister chromatids. Sister chromatids are each one of these chromosomes being duplicated and still adjoined. So every chromosome from my mom has been duplicated at the start of meiosis so I should have said first, interphase is the same. So meiosis begins with interphase. So we've already gone through interphase, which includes S phase, the duplication of each chromosome. So when we start out meiosis, we're starting out with duplicated chromosomes. So every single one of my mom's chromosomes were already duplicated during interphase, in S phase specifically, and every single one of my dad's chromosomes was duplicated in S phase. So you can see here that this cell is diploid right here. So meiosis in general is the production of haploid sex cells. But we start with diploid cells. We start meiosis with, basically somatic cells, cells that are destined to become sex cells. So here, let's put this here. This is a diploid. This cell is diploid, and each chromosome is also duplicated. They were duplicated in interphase during S phase. So these two chromosomes are homologous. This is a homologous pair. That's why I've drawn them to look the same. They are not identical because my dad and my mom might provide me with different versions of each gene, but they're homologous because these two chromosomes, the one from my mom and the one from my dad, they include the same sets of genes. You know, like if all eight genes for eye color are on these chromosomes, then they're on this one from my mom and on this one from my dad. They're homologous in that way. They have the same sets of genes in the same loci, but they're not identical because my dad might have given me different versions for the eye color gene than the ones that my mom gave me. So maybe my dad gave me a whole bunch of genes that code for brown eyes, and my mom gave me a whole bunch of genes that code for blue eyes, right? So the inheritance chapter in chapter 9 tells you like what happens when you get conflicting codes from mom and dad. Those are called different alleles. If my dad might give me alleles for brown eyes, my mom might give me alleles for blue eyes. So they're not identical. They're different codes. There's mutations that have happened so that I can get different versions of a gene. But they're homologous because they're for the same feature, eyes. Right. All right, so this is a homologous pair, and each homologous pair is made out of two duplicated chromosomes. This is a homologous pair. This is a homologous pair. Right. So sex cells have to be haploid because remember the whole point of having sex, sexual reproduction, not of having sex, right? We have sex for lots of reasons, but the whole point of sexual reproduction when you're making a baby via sex is to join a father's genetic information with a mother's genetic information and recombine them to make a whole new person. So if I want to mix my genetic information with another person's, my husband's genetic information, I have to cut my genetic information in half. And he has to cut his genetic information in half because then when you combine a half with a half, you get a whole again, right? So we want our kid to also be diploid, not tetroid. We don't want our son, if I didn't cut my genetic information in half and my husband didn't cut his genetic information in half to make our sex cells, then when our sex cells fused, our kid would have, instead of being diploid, instead of having two copies, you know, one set from each parent, our kid would have two sets of genetic information from each parent, right? A diploid cell plus a diploid cell, our son would have 4 copies of every chromosome. And then his offspring would have 8 copies of every chromosome, and it would just keep collecting. You get too much DNA in the cell by the end of just a few generations. So we want to have our son have the same amount of DNA as I have and as my husband has. So I cut my DNA genetic information in half, my husband cuts his genetic information in half, and then we put it together. And a half plus a half makes one whole, right? So our son will have the same amount of genes as we have. So the way it's not just a random assortment. You can't I'm not just taking a random half of my genes and putting it in my eggs. I get one full, every one of my cells has 2 full sets of genetic information. I'm going to take 1 full set of genetic information. So it's not like I give some eye genes but no nose genes. I get 1 copy of each eye gene goes to each of my eggs. 1 copy of each nose gene goes to my eggs. 1 copy of each hair gene goes to my eggs. So I have 2 of each gene. I have 2 of each eye gene. I have 2 of each hair gene. I have 2 of each nose gene. But I'm taking 1 of each to put to my eggs. So each of my eggs has 1 full set instead of 2 full sets. So 1 full set of genetic information is haploid. 2 full sets of genetic information is diploid. So let's just see how this plays out. So humans have 46 chromosomes. I'm not drawing 46 chromosomes. That's why we work with a yellow fever mosquito. Yellow fever mosquitoes only have 6 chromosomes. And we call them, and they're also diploid. So we say to write how many chromosomes we have, we say 2n equals 6. The 2n means that it's a diploid cell. There's 2 copies of each chromosome. 1 set from mom and 1 set from dad. And the total number of chromosomes is 6. So this is 2n equals 6. There's 6 chromosomes, right? That's where the 6 comes from. 1, 2, 3, 4, 5, 6. But 1 set comes from mom and 1 set comes from dad. That 6 is composed of 2 different sets of 3. So that's where the 2n comes in. If it was just n equals 6, that would mean that it's a haploid cell, like a sex cell with 6 chromosomes. But that'll become more clear later. So we're starting with 2n equals 6, a diploid cell with duplicated chromosomes. And this cell is destined to become egg cells for this yellow fever mosquito. All right, so this is just an overview. I'm not going through each step by step. I'm just doing an overview of the point of each stage of meiosis. So the purpose of meiosis 1 is to separate homologous pairs. That's the whole point. So you go through prophase, metaphase, anaphase, telophase. Prophase, chromosomes condense, just like in mitosis. In metaphase, they line up along the equator, but they line up, well, we'll look at pictures of that later. The homologous pairs line up on the equator. And then anaphase, they separate from each other. In telophase, nuclei reform, and then in cytokinesis, the cells separate. So at the end of meiosis 1, so this is just the overview, we'll look at all of that in just a second. But at the end of meiosis 1, we have, oops, let me see what I've done here. Here we go. Oh no, that is not the color that I want. Maybe I wanted this. Aha, that's what I want. Oh, that was weird. So the genetic information in meiosis 1 also gets mixed up a little bit. So we're going to have a mix of dad and mom's chromosomes in these two different cells. Black here, push this off. So you can see that mom and dad's chromosomes got shuffled before they separated. And that's an important thing that happens in meiosis 1 as well. So the purpose, so here, let's see, shuffle maternal and paternal chromosomes. Okay. That's something important that happens in meiosis 1 also. So in meiosis 1, you separate the first you shuffle the maternal and paternal chromosomes, and then you separate the homologous pairs. So you can see here that each daughter cell is still made up, each chromosome is still duplicated. It's still made up of 2 sister chromatids. But mom and dad's genetic information have separated so that each daughter cell has 1 of each homologous pair. So these 2 homologous pairs separated to different daughter cells, these 2 homologous pairs separated to different daughter cells, and these 2 homologous pairs separated to different daughter cells. You'll also notice that not only did these homologous pairs separate, but they actually switched, little pieces of each one popped off and switched places with each other, right? So this one's arm, an arm came off of one and switched places with the other. Right? So that's called, so we'll talk about all of that when we get into the nitty-gritty. But that's part of the shuffling of maternal and paternal chromosomes. So now each daughter cell has 1 of each chromosome. That's important because each daughter cell, each sex cell has to have 1 complete set of genetic information. So it's not like you get like you don't just split up any 3. You can't take this one and these 2 to go to one daughter cell, because then that daughter cell will have too many of these chromosomes and none of these chromosomes. That will just make some kind of deformed, non-viable offspring. So if meiosis is happening correctly, homologous pairs separate so that 1 of each kind of chromosome goes to each daughter cell. And you also have a shuffling of the maternal and paternal chromosomes, and that's important because that's where the genetic diversity comes from. That's why every sex cell is unique. All right, and then in meiosis 2, the important thing that happens in meiosis 2 is, I'm going to leave myself a lot of space, but so separate sister chromatids. Separate the sister chromatids. That's the whole point of meiosis 2. Meiosis 2 is really just, I can't move that up anymore. Okay. Meiosis 2 is really just mitosis. You're just separating these sister chromatids into 2 daughter cells. So if I were to draw out where these things were going. This cell splits into two here. Sister chromatids split up. And again, it goes through prophase, metaphase, anaphase, telophase. Prophase, the chromosomes condense again. Metaphase, they line up on the equator. Anaphase, the sister chromatids separate. And telophase, nuclei reform. So prophase is always about condensing of the chromosomes and dissolving the nuclear envelope. Metaphase is always about lining up the equator. Anaphase is always about separating something. And telophase is always about reforming nuclei. Cytokinesis is always about the cell dividing into two around the two new nuclei. So let's see what this is going to look like. So if I were going to separate that genetic information, so remember, now they're not duplicated chromosomes anymore. Oh, that blue didn't. There we go. So in these, I've split up the sister chromatids. So I split up this sister chromatid from that one, this one from that one, and this one from that one to the two daughter cells. See how that worked out? And then this one I'm going to do the same. So am I on blue? So this sister chromatid splits and one of each goes to each daughter cell. These two sister chromatids will split. One of each will go to each daughter cell. Oops, let me make it the wavy kind so you can tell the difference. I'll make this one the one that has a little bit of a mix of paternal and maternal information. The wavy one, and then there's the little one. That little one also gets split and one of each goes to each daughter cell. So now, each daughter cell still has, so this is the end, meiosis II is the end. Each daughter cell has one of each chromosome. See, there's the long one, the wavy one, the short one. Long, wavy, short. Long, wavy, wavy, short. Long, wavy, short. Every daughter cell has one of each chromosome, one complete set of genetic information. And they're all also unique. They have, each one has a unique combination of this mosquito's mother's and father's genetic information, so that each mosquito egg is genetically unique from every other mosquito egg. And this is also how sperm are created. So it's the same process. So I said we started with a diploid cell with duplicated chromosomes. At the end of meiosis I, these cells, these daughter cells are haploid. N equals 3. So the 3 represents that there's one of each chromosome, there's 3 chromosomes total in each of these cells. There's 3 chromosomes, 1, 2, 3. And N, there's not a 2N, N it means it's haploid. So there's not 2 of each type of chromosome. There's just 1 of each kind of chromosome. So that's N. And when there was 2 of each kind of chromosomes, you had 2N. All right, and then you have, so let's write this out. You have, this is a haploid cell, but the chromosomes are still duplicated. That's true for both of these daughter cells. And actually, just so this looks more clear, I'll draw some arrows so you can see that these are the daughter cells. Okay, so each of these are haploid now, but they're still considered to be duplicated chromosomes, right? These are dyads, to use the technical terms from the book. And then here you have, each of these is also N equals 3. So that hasn't changed. The only thing that's changed is that instead of each of these chromosomes being duplicated, now each of these chromosomes is unduplicated. I'm running out of space here. Let's see. Haploid, unduplicated. Okay. All right. So that's meiosis. You start out with just like a somatic cell that is destined to become sex cells, and it undergoes 2 rounds of cell division. The first round of cell division shuffles maternal and paternal sets of genetic information to make genetically unique daughter cells, and also separates the homologous pairs to make haploid cells. Meiosis II is only about separating the sister chromatids. And then at the end of meiosis, you have sex cells that are haploid and that are all genetically unique. That's why we are all unique snowflakes. In the next lecture, I'm mainly going to talk about the shuffling that happens. And then we'll just go briefly through the PowerPoints of all the other stages of meiosis.